Ladies and gentlemen, welcome to the stream. Everybody for joining. It's been a couple of years and excited to start this again. Have another great presentation today. Dr. Kyle Grice, DePaul University, about mistakes in science and literature. Try and fix them. And so, for anybody who might be new, first of all, welcome to us. Excited to have you here. And the goal here is to basically provide a place for virtual format, but it enables a wider audience to be questions absolutely encouraged. Got a couple of different. First is Discord. I have a Discord server that's set up, handling there. Got a program that automatically collect them. Working exclamation option no longer works, but basically, if you ask questions, you can still use the exclamation question. Have any other just general questions about anything? Got the whole this talk and adding my own questions. So absolutely. Feel gotten something, but I think that pretty much covers it all. With that, I will say again, Kyle Grice, DePaul University. Kyle, please take it away.
All right, thank you. Thanks, Ben. This is awesome to be back. I've actually given 2 of these talks before. And again, folks watching, let us know if there's any audio or visual issues. But so let's get going. All right, so a little about me. I'm a professor at DePaul University. I'm a chemistry professor. And I've been there for 12-ish years. If you're ever come through Chicago, come say hi. We've got 2 campuses, one in the Lincoln Park campus, LPC, and that's where I am. It's actually, if you know Chicago a little bit, the Lincoln Park Zoo, Wrigley Field, right? And if you draw a straight line down from Wrigley Field, we're down here. So there's also a Loop campus, and that's other parts of the university. I'm not down there, though. Anyway, we're undergraduate, primarily undergraduate. We have a small master's program, no PhDs or postdocs, but we do lots of research with undergrads. We're on the quarter system, which is a bit different than many schools. So I'm starting my quarter up here in September 10th. And there is a DePaul in Indiana. So yeah. One thing I want to say to the chemistry folks too, if you're an American chemicals person, the American Chemical Society meeting in the fall, which is really in August 2026, is in Chicago. So I hope to see lots and lots of folks there. I'm certainly going to go there and take as many students as possible. All right, so a little bit more about my own research. I'm an inorganic chemist by training, but I've branched out a lot. A big area of my research is turning carbon dioxide into something useful, and one of the stories I'll talk about today kind of comes from that area. And then I'm also interested in platinum chemistry because that was my PhD. So we study platinum complexes still. And then I do lots of bio-inorganic chemistry and organic chemistry even too. So I do a lot. And I'm also thinking about green chemistry and how to do things more green or renewable. And so that's been a lot of fun. But yeah, so if you want to see some more about my research, I've actually gave, like I said, 2 talks previously. And so they're on the YouTube channel. All right, let's talk about the topic of the day, mistakes. So really, I think sometimes students especially don't think about science as being something that had lots of mistakes in it, but it does, right? We make mistakes all the time, we're humans, and we learn from those mistakes. And so you mess up pretty bad, and then okay, I'm not doing that again, and you learn and you can. It's a powerful learning strategy. And so, and this is this makes sense, right? If you think about it, in lecture classes, the best way to learn is to do practice problems, and it's okay to get them wrong. You got to check your answer and see what you got wrong, and then fix it, right? So that's kind of a really normal way of learning. Now, science and doing science, right, is a human thing. We make mistakes. So mistakes get into the scientific literature. It happens, right? And so the issue, or the nice thing about science, is if you're doing it right, Science should be reproducible. And so scientific literature builds on previous work. And so that, in a way, can correct mistakes. I'll show some examples of that. Like, if an initial paper is wrong in some way, and then like it gets corrected and so on. But also like just learning about these mistakes, right, is useful because you can learn about like, okay, what went wrong, and how do we not make those same mistakes in the future? Like what interpretation was wrong? What did the data say that was actually not what it was saying, etc. Okay. Now, there's many types of mistakes, and so I'm only going to talk about a certain type today, but there's all sorts of things, right? So like mistakes can be little tiny things like a typo. They can be big mistakes, like something can be totally wrong in terms of the full, scientific basis. But mistakes can be everywhere. It could be in raw data, or like a processing issue. A lot of mistakes I'm going to talk about today are like interpretations of data, and mistakes get made, or like, even like making figures, you can get stuff. And so there's all sorts of mistakes. My assumption is that most mistakes are honest mistakes, like, not on purpose, right? Not fraud, but fraud exists. And I'll mention that a little bit. But one thing that is interesting about this whole area, and I think why people don't like to talk about it too much, is that, people's egos get involved with their science, right? Like you want to defend your science against other people. And so sometimes it's hard to let go of your, incorrect assumptions or interpretations. And so it can be pretty surprising. All right. So what's the big deal? Everybody makes mistakes. Meh. Maybe we shouldn't care so much about trying to fix it. Well, here's where I think it's important. Right? Mistakes in the literature propagate. Right? We cite previous things, and then so people cite previous ideas that might be incorrect. And that's kind of how science goes, anyways. Like people make the best assumptions or interpretations they can with the tools they have, and then it gets corrected as we go. But some of these wrong ideas can kind of get entrenched, right? And then people use these papers, the data for grant applications, patents, investments to startup companies, et cetera. It could all end up be something incorrect, right? They also end up in our classrooms, right? So we're teaching students these incorrect things. If you look at like old textbooks, right, they teach some wacky things that we know are wrong now. But so there probably are some things in our textbooks now that are also wrong, and that will maybe be fixed as we get better interpretations. Okay, so that's, again, kind of just some text, but some people might say, well, what about AI? It'll just like kind of fix everything, and like, and we don't have to worry about it. Well, no, AI is trained on data, right? And so if the data is incorrect, then the AI will be incorrect. And this is a great blog post from Chemistry World, which is kind of the blog of the, or like kind of like magazine of the Royal Society, I think, and it's kind of like the equivalent to like CNN News for chemist ACS. But this is a story about how, and it cites some papers about how people found out that pKa values they were using from chemical databases for like AI training were wrong. So the raw data was wrong. And there's an old saying in chemistry, and this was usually for synthesis, like garbage in, garbage out, right? If your starting materials are dirty or not pure, or you don't know that they are what they are, you're not going to get good results. So, this is true for synthesis, right? Making stuff, but it's also true for just thinking, right? Like, and the garbage data in, you're going to get garbage data out. So, this is why it's also important in the growing world of AI is we need to make sure that we're training our machine learning on good data sets. Yeah. Okay. So, ideally, and sorry for all the text, I'm not a super creative, artistic person. I promise there will be more figures on subsequent slides. Okay, so ideally, this would not get into the literature in the first place. So how can we do that, right? Well, remember that if science isn't reproducible, it's not science, right? So double check everything, reproduce your experiments. Be so clear in your methods that have other people try to reproduce them. I'm working on something right now where like we reproduced it a few times, but I want to be absolutely sure. So I asked a collaborator at a different university and be like, hey, can you do this, and then get your data and let's talk and see you can compare, right? Challenge your own assumptions, right? We have our knowledge is built on these assumptions, and maybe not all of them are wrong, are right. Sorry. Look at the data. Be critical. Another great thing, and this is what I miss from being in a research group in like grad school and postdocs, is like during group meetings you would do a practice talk before your presentations, where you have other people read your work. It's really important. So many times I've been writing something where like I think I'm saying something, but I'm not, right? And so have people read. The other thing is peer review, while it has flaws, is valuable. And so peer-reviewed manuscripts are better, can be trusted better than non-peer-reviewed, but there can be errors. And so I encourage everyone to be a good reviewer, right? So that means be thorough and critical, but supportive, right? Critical of the data and how it's being written, but not a jerk, right? And then also don't use AI for this. Do it yourself. Be a scientist, right? The other thing is, now that most papers are online, it's easier to put lots more pages in there, right? So like you can put your raw data, spectra, pictures of things, of how your setup was done in your supporting info. And more journals or articles are doing this, which is great. Just making them exceedingly clear of how you did something, right? Finally, and I'll talk about this in a second, when you're processing images and data to make it look good, don't overprocess them, right? Don't exclude data, don't hide things, right? That approach is fraud. And that's something that is not really the kind of mistakes I'll be talking about today, but is certainly a big problem. And this woman named Elizabeth Bick is doing great work in that area, and there's plenty of others. Now, before I go any further, I am also a human and make tons and tons of mistakes. There's probably mistakes in this talk, like in terms of typos and errors. But I've made so many mistakes. I try to teach my students the mistakes I've made so that they can learn from them. They can be like math mistakes, right? Like doing calculations wrong or assuming something when really it's not going to do that. That happens all the time. That's like basically research. Or trying to do things quickly and like not writing down everything. If it's not clearly recorded in your notebook, basically it's not science. It never happened. And then typos get into places. And again, this is why it's always a good idea to have folks look at your talks and your papers before you do the final thing. I didn't have someone look at this talk, so there's probably mistakes. I also have broken so much glassware. All right, breaking things is not a mistake in my mind. That's normal. This is part of the work that we do. Students, the first time they drop something and panic and like, no, it's okay. Like I break glassware all the time. Sometimes I'm like showing a student something, it slips out of my hand and I'm like, oh, well, there goes that. Anyway, so let's get a little further in here now. Okay, I'm not talking about, and the case studies I'm going to talk about start in a couple slides here. I'm not talking about outright fraud, which is knowingly faking data, images, etc. That's not part of the stories today, at least as far as I know. But it does happen and it's worth talking about. And there's lots of famous examples in all fields. There's always somebody who just gets desperate or greedy or, starts just breaking rules and getting away with things. And then it builds up and finally comes crashing down. For example, the company Theranos and the fraud there. There's a good book called Bad Blood that details that whole story. When money is involved, it seems it's more likely sometimes that can happen. So speaking of that, people are doing great work looking at the literature and trying to fix it afterwards. And I'm not doing that a lot, but Elizabeth Bick is fantastic. She does that. There's also a great website that tracks retractions as a way to understand science when like a paper gets removed from the literature. Further, I'm not talking about like injuries, accidents, spills, etc., which are also important. That's like safety. And you can spend talks and talks on that, but I'm not going to talk about like, chemical spills and like accidents or deaths and injuries, but they happen and you should know about them. Okay. So just a couple quick things. If you haven't checked out Retraction Watch, please do. It's pretty cool. It's like a little simple website where here it's just like a news feed. They keep track of retractions and like try to figure out why they happened and what's going on and things like that. But yeah, it's just like tracking retractions as a window into the scientific process. I mentioned Elizabeth Bick, and there's other people besides her, and they're doing great work. I don't know them. I haven't met them in person, but I've been seeing what they do on social media. So this is her social media thing. She's got a Wikipedia page. Mu Yang's another one who's I see on and others on social media kind of looking at published data. And one way to do that is a website called PubPeer, and that allows people to post comments about papers that are published, and sometimes anonymously because you might be afraid of, what's the word, retaliation, right? But yeah, it's a useful way for people to point out and say, hey, that figure looks weird and stuff, right? And I haven't used it much, but again, Elizabeth Bick, Mu Yang, those folks use it a lot. So I encourage you to check that out. All right. So I promised I would talk about some stuff today besides just generic things. So let's get into some details here. Let's talk about 3 case studies that I've seen. One of which, 2 of which, these last 2 I have been working on publications related to. The first one I was kind of like in the sidelines watching it all happen. And there's plenty of other stories out there. You can look up some other ones for chemistry. There's something called the oxo wall. It's about if metals can have oxos or not. And there's several cases where people thought they could make oxos with metals that don't have oxos, but then the data was interpreted wrong. There's also, the physics and material science world is trying to make room temperature superconductors. And there's been some kind of mistakes and fraud and stuff in there. And then like biology, there was, I think a paper finally got retracted that was on arsenic-based life instead of phosphorus-based life. But anyways, tons of examples, but let's talk about these 3. All right. So a little background for case 1. So many people, I do CO2 reduction, right? This is where so in my postdoc is when I started learning about CO2 reduction. And that's when I learned about this problem. So CO2 is in the atmosphere, it's increasing the air, it's bad, it causes ocean acidification and climate change. So we should do something about it. There's all sorts of ways of trying to tackle this problem, from making less CO2 to capturing CO2 and sticking it in the ground to, what I'm interested in is recycling CO2 into useful things, like doing chemistry with CO2 to make things, right? So it's a viable kind of starting material. An example of this would be like, copolymerizing CO2 and epoxides to make plastics, polycarbonates. I look at CO2 reduction. So CO2 is in carbon plus 4, the most oxidized form of carbon. It's the product of combustion. You can reduce it by 2 electrons, either to formate in the presence of a proton, or basically this is a hydride transfer, or you can also reduce it in the presence of proton sources to CO. But both of these are 2 electron reductions. And those are the most common reduced forms for most catalysts I've worked with or read about. Ideally, you'd want to reduce it all the way to a liquid fuel that like methanol, but that's a 6 electron reduction. So methanol has always been kind of a big target for CO2 reduction folks because, hey, it's a liquid fuel, very similar to gasoline already, right? Like it's a liquid you could put in the gas tank and burn. It also can be used in fuel cells too, right? So that's been a big target. All right, so I started my postdoc right after my PhD. I got my PhD in 2010, right? And I was doing platinum chemistry and just like doing one thing by myself at University of Washington. I wasn't, in a collaboration. I was, just in the lab doing stuff. And so then it was a big switch for me going to the Kubiak lab for my postdoc, which was a big collaboration project I was on. I was leading it, kind of like organizing the group meetings between all these groups. We were looking at CO2 reduction homogeneously, heterogeneously, theory, experiment, all approaches. And so I learned a lot. It was great. And that's why I continued to do CO2 and saw the value of collaboration. And that's when I saw this kind of fiasco and debate and issue play out. Okay, so before I joined that grant, the MURI, these 2 papers were published. And it's from the group of Bocarsley and Princeton. The first one here is a semiconductor surface called p-doped gallium phosphide. And they found that using a pyridine-pyridinium aqueous buffer with light and electrolysis, you could actually make methanol. And that was really exciting, right? You can make methanol from CO2. And then a few years later in JAX, they published the same thing, but just at platinum. So not photochemically, just electrochemically. And they saw this beautiful, what looks like a reversible signal at negative 0.6 volts versus SCE for reducing pyridinium cation to pyridinyl radical. And then they proposed this mechanism where pyridinyl radical attacks CO2 and you form these things and it goes all the way to methanol. All right. So this was at a platinum electrode. Okay. So literally all you need is a platinum electrode, some pyridine, some acid and CO2, and you can make methanol, supposedly. But some problems. Everybody got really excited about this, right? Because it seems so simple and so cool, right? You could just make methanol just by adding a small organic base or acid. And so people tried to reproduce it and were having trouble. They could see that reduction, but they couldn't make the methanol that Bocarsley was saying. People looked for that radical and could not observe it. Right? If it was a radical that was stable, which they claimed because of the reversible looking echem, you should be able to see that species by normal radical techniques like electron paramagnetic resonance is a tool for detecting and studying things that are radicals that have an unpaired electron. So that this is, you know, an issue because everybody's trying to reproduce it. And this was mainly through, like, people hadn't published stuff, but they were working on it, and they would talk at conferences and argue and complain. And so it came kind of, people began to realize like something's not quite right. A mistake is being made somewhere is it either the people trying to reproduce it or is it the original data? And there's a whole slew of literature out there on this, people trying it and then publishing stuff on it, people trying computational to try to find the mechanism better, people doing electrochemistry to try to double check it. And so I'll mention a couple below. All right, so again, 2008, 2010 is when the pyridinium papers were published. I started in that collaboration in 2011, so I was just after all of that had been published. So in the same collaboration that we were all in, Bo Carsley, Kubiak, and then there was Emily Carter as a computational chemist, and she had a great postdoc, John Keith, who's a friend of mine. He and I collaborated and co-published papers back then, and also we've co-published papers since then in my own career. But I wanted to, you know, these were his postdoc work. And so what he did is he's like, okay, I should be able to calculate the pyridinial radical. He should also be able to calculate the reduction potential and try to calculate the mechanism. And so he did extensive benchmarking and published that on pKa's and reduction potentials. And then he calculated the pyridinium reduction, right? It's supposed to be a homogeneous catalyst. This is its observed reduction potential. This is his calculated one in water. It's about a volt off, which is a pretty big difference. If you don't know, a volt of energy is significant. It's not like a small energy difference. And so that was disconcerting. And so he calculated a bunch of other things to like benchmark. And his benchmarking is pretty good. This is, I think, in acetonitrile actually. And even then, people had measured it in acetonitrile and seeing the same thing. And but again, it was calculated way off. But other things seem to match, right? Other things seem to match literature data. So something's up with the original thing. So basically what this DFT was saying, his calculation saying is that observed signal cannot be the reduction of homogeneous pyridinium, right? So I remember the group meetings that this was discussed, and I was not at Princeton, but John Keith was, and there were some yelling matches and arguments, heated arguments, but eventually, you know, I mean, I don't think you can refute this data. And so Carsley, you know, there was lots of arguments. I saw some crazy ACS meeting presentations where every speaker was kind of basically like, you know, refuting the previous speaker all up on this topic. But Carsley eventually came around because he realized that, hey, it wasn't just pyridinium on platinum. Any weak acid in water on platinum shows a signal in the same region and is pKa dependent. It's basically like a pH electrode. And so you can basically measure the pKa of acids with platinum, which because it's been done before, right? And so I've looked at this too in water and acetonitrile, and yeah, you can see that signal. But so this suggests that pyridinium reduction is really just proton reduction at platinum. And again, this is in early 2010s, right? And so many other people were publishing. And so this is what I would consider maybe the key kind of final electrochemical paper. And I spelled Konstantin's name wrong in the title. Sorry, I apologize. They're great chemists. Konstantin Saviant is retired. Konstantin is still doing great work too. I think he trained with Saviant. They're from France. I'm sorry, I'm probably butchering their names. Again, I apologize. But Saviant's work earlier and Konstantin's work continued more recently are all great work and really good electrochemistry stuff. They host a workshop on EChem and cyclic voltammetry too. So like they are great people. And so when they do work, you know it's going to be great. And they did a very thorough and extensive paper, and they confirm, or they kind of support the idea that yes, what's happening is, you know, it's just a weak acid. And then the H plus forms a surface hydride. So even though it looks homogeneous, it's actually heterogeneous, and they figured that out as well. And then some reduction does happen, but it's very minor. And it's mainly outcompeted by hydrogen production. So, you know, this was, again, kind of what I would consider the nail in the coffin experimentally on this idea. But it was a pretty big discussion in the literature, lots of big papers, lots of JAX papers about this. Okay, so just to show you that I've done this, and yes, I agree. So like you can just get pyridinium, right, in some water, and you can bubble CO2 into it. I like to use an internal reference, and so for water, I use this ferrocene methanol, which is a water-soluble internal reference, because sometimes reference electrodes drift or do weird things. Something gets stuck on the surface of your reference electrode and changes its voltage. So it's always good to have an internal reference. And sure enough, at glassy carbon, which is an inert electrode, the inert electrode of choice for electrochemists, it used to be mercury and platinum, but now it's glassy carbon. And you can see a peak down here at about negative 1.5 volts just as you approach the solvent window on the reduction side of water. And it increases under CO2, right, because you're making more of the protonated species. And it's about negative 1.5 volts versus the SCE and DFT. My DFT and John Keith's was about, his was about 1.4. I did some DFT too and got about 1.6, negative 1.6. Yeah, we're close. Like I'm consistent. I think this is it. If you switch the same solution to a platinum electrode, you see a bunch of other stuff earlier. You can't even get that far negative because you just start making hydrogen, which platinum is great at. So yeah, here's these signals, or at least one or two signals of weak acids. This is probably bicarbonate, and then this is the pyridinium weak acid proton reduction at platinum. So this is fundamentally an error of interpretation, a mistake of interpreting data, right? And I thought that the story had kind of been ended, but then when I was reading some literature, I saw this paper, and they missed the memo. This is 2023. It's a photochemical organocatalytic paper. This is ignoring all of this, I just want to point out that their mechanism involves reducing pyridinium to pyridinyl radical, which they do electrochemically, and they see it at negative 0.6 volts in acetonitrile, because they're doing this in acetonitrile, which again, that's not this. This is just weak proton reduction. The good news is if you read about the mechanism in this paper, the reduction potential of their reductant is strong enough to reduce the negative 1.56 volts or negative 1.5, negative 1.6 volts of this anyway. So it's got enough driving force. It doesn't really change their mechanism, but this is a wrong number. That's not pyridinium. Now, again, I've done this and I've had students in my lab do this. So a master's student in my lab, Josh Lutke, showed that if you take a platinum electrode and acetonitrile, you can measure pKa's in acetonitrile, which have been measured. And again, in acetonitrile, on platinum, it's about negative 0.6 volts. It's like this, but again, that's not pyridinium. That's the protons being reduced by a weak acid at the surface of platinum. And this is voltage versus pKa, and it's roughly linear, not perfect, but roughly linear. This is a bit more of his work from his thesis. You can access it in DePaul's library. He tried these acids in acetonitrile with these known pKa's, and he did averages and stuff. And here's all the signals versus ferrocene as our reference. Different peaks here he's measuring. And because they're irreversible, which is something in electrochemistry, we tried measuring it a few different ways, and it turns out that it's best to measure the half height location to estimate. But basically, you measure a pKa of a weak acid and acetonitrile on platinum, which is kind of cool. Anyway, so the summary here is that from this case study is that, right, pyridinium and weak acids get reduced to platinum, but that's not a homogeneous thing. Also, the pyridinyl radical is not a stable thing in solution. You can observe it in wacky, like matrix isolated things, right? But you can't really observe it in like room temperature solution. That CO2 to reduction to methanol is probably complex and heterogeneous. Bocarzy's early work, those two papers he was running, there's a potential galvanostatically, which if you're an electrochemist, you know what that means. It means that he didn't say run at this voltage. He said, give me this much current. And then it moved the voltage around to give that. And he didn't report what the currents were, the voltages were at when he got the methanol. So we don't know, right? We can't repeat that experiment because we don't have all the details. The central mistake here is interpreting that heterogeneous reduction on platinum is a homogeneous one. That's kind of like the central, it's a data interpretation mistake. The data is real, but the interpretation is wrong. Okay, so some lessons. For everybody, platinum is not an inert electrode. Don't use it as an inert electrode, even if other people are. Use glassy carbon. Galvanostatic electrolysis, unless you want to do that, it's going to move the voltage around. But so report that. If you're going to report the galvanostatic electrolysis, show what voltages it was running at. I prefer to do controlled potential electrolysis, where you define the voltage and you hold it there. Also, read the literature widely. A lot of stuff has been done out there, sometimes refuting papers, and people don't recognize it. I always recommend forward searching your articles, like seeing what cites them, to see if something's been refuted. Like you find a cool paper, maybe it was wrong. Also, before proposing stable radicals or wacky structures, be sure to get lots of supporting evidence and rule out other options. So for example, we can now say unsubstituted again, that pyridyl radical is not stable in solution. And finally, like back when I was in my post and my grad school and earlier, some people were said like, oh, DFT is not very accurate. It's good for kind of trends, but you got to be careful actually. But I think it's getting pretty darn accurate in some cases. So it's very useful, but be sure to benchmark it. I'm now doing DFT on lots of things when I didn't, it's a tool that we can use, even people who were trained experimentally like me. All right, so that's the first big thing I saw, and that was an interesting thing to be on the sidelines of, and dabble in a little bit. I didn't publish any papers on it, but yeah, it was pretty crazy. Okay, case study 2. This is more recently. This all started with a blog post I read from the Baran Lab blog. It's defunct in that I don't think they posted anything recently in the last several years, but in this, they saw a paper that was a couple, one paper or two papers, and then it was cited in a review with this structure. And they said, this is bananas. There's no way this can exist. It's probably this. And they even went and took the mass spec and proton NMR and carbon NMR of this thing, and it matched pretty well with the data from the papers. And so I love this quote from the blog post authors. Our conclusion, if something looks really unusual, check all the reasonable hypotheses before you propose something exotic. Sometimes things are zebras, but if you hear hoofbeats, it's probably a horse. All right. So, and I saw this and, I was thinking about it and I was, showing folks, and I had a master's student who was interested in computational chemistry. And so I thought, well, how else can we study this? We could do computational chemistry. And so the student, Nick Ching, and I worked together and calculated a bunch of things using DFT. And so we published it in Tetrahedron Letters. And so if you want to go read it, this is the paper. But the bottom line is, yes, this is bananas and cannot exist. When we tried to DFT optimize this structure, instead we get this, which is a very, very bad ring strain structure of benzene. You've broken the aromaticity. You've got 2 4-membered rings with insane amounts of ring strain. But these things do actually, this type of structure is known. It's called a Dewar benzene, but they're insanely reactive, right? And I tried to stretch that middle CC bond in DFT by just basically elongating it and optimizing and elongating and optimizing. For those of you who know DFT, like it's called a relaxed scan, right? And it kept crashing. I could not get anything like this. So what did we do? Well, we said, okay, well, we know we can optimize a Dewar benzene. How unstable is it? So we calculate it compared to benzene. And then this thing, well, let's calculate it compared to something that we know exists. So this is something you can get. It's an oil. It's air stable, right? And these numbers basically tell you in kilocalories per mole, this is ridiculously uphill and high in energy. These Dewar benzene things are super sensitive to air, water, and temperature. They're isolated in situ. They can react with everything nearly. Very touchy. And we compared the oxygen ring versus the carbon ring, like the 3 carbons here versus an oxygen and 2 carbons. And the 3 carbons is actually not as bad, but still pretty bad, right? And so we looked in the literature, and there's these things called paracyclophanes that do exist with long linkers, right? But then there's the orthoisomers, which are more commonly known, like this guy and this guy. These are stable, right? So we calculated a bunch of these and compared and with these. And so we had 2 reactions here, this going to this or this going to this, right? And then we started to plot the energies of these things. So here's a fun little plot. So this is the number of carbons in that bridging linker in an attempted paracyclophane. And there's 3 species here. The blue is the more stable orthoisomer, the things that are known you can buy in a bottle. The black are the duobenzenes, right? Those really strained benzene things. And then the red are those linker things. So we couldn't even calculate one with 3 linkers, 3 atoms. We could with the 4 and the 5 and the 6, and becomes more stable the longer the linker is because there's less ring strain. And you can, these are known. The 6-member one is published and it's relatively stable. The 4 and the 5 are so reactive they fall apart at low temperature, right? And it's not until you get a ring 8 linkers from in the para positions that it actually becomes about as stable as the orthoisomer. So basically, again, duobenzenes are ridiculously high in energy. These paracyclophane things are high in energy unless you have a long linker, right? A long group. Now remember, the thing that they were proposing had a 3 linker, so the energy would be somewhere up here. And this is just for the all carbon ones. So let's do those oxygen ones too. So here's with the oxygen in it. Now remember, the oxygen actually is, CO bond's actually shorter than a CC bond, or can cause, ring strain. And sure enough, it's even higher differences. It's even higher in energy compared to the stable orthoisomers than the all carbon ones. So again, these things are ridiculously unstable. You cannot make the stuff, the thing they're proposing. You could even look at this ring strain in the benzene, like how bent out a plane it is. We did that, right? And so on and so forth. So it was fun. A fun kind of computational study. Further, you can also just use like chemical intuition, right? So this thing should be an oil because it's got no hydrogen bonding things, but they isolate it as a solid after doing 2 methanol columns in air. So it's got to be air and water stable. And so the blog post proposed this, and that they think the mass spec they were seeing was really just a dehydration of this, which has the same mass as this, but they have different masses. And so if you isolated the crystals and you sent it off for elemental analysis, you could see how different the carbon percentage is. I'm not a fan of high-res mass spec. Organic chemists do that for saying something is pure, but really you're only saying that compound that I think I want is there. It doesn't say bulk purity. Elemental analysis, burning it, does do bulk purity, but it has its own problems too. I mean, again, this is the importance of trying multiple different techniques. And I searched through SciFinder trying to find anything that looked like this. And then I added another carbon and then searched again. Add another carbon and searched again. And it wasn't, this was the shortest like ring linker I could find. And this was not crystallized either. This was NMR. But I calculated the structure and it's about the same as the 6 linker species I calculated. So, right, again, you cannot do those, have this ridiculous structure. So yeah, again, lessons, right? I agree with the blog conclusions, but read the literature widely. Again, if the authors of that paper knew that these paracyclophane things were so reactive and not stable to air and water, they would not propose that structure. And again, if we had seen the raw data, we could maybe talk about it better. Like they took an IR, but they didn't report the OH groups. And maybe they were there, they just didn't think they were peaks, right? And then here also, again, computational chemistry can be a powerful tool to say, like, is this even possible or not? But be careful, again, you got to benchmark, be careful about your tools, talk to collaborators, colleagues, and get to know. Make sure that you're sure what you're doing is legit. But yeah, so before you propose something new, make sure you're absolutely sure it is what you think it is. All right. Okay, so the last little quick thing I want to talk about today, and we can talk forever about these types of things, but I want to get back to the idea of protons and pKa's. And I mentioned that Chemistry World blog post and AI training article early on. So protonation sites and pKa's are very important to know, right? You need to know the protonation state of your compound in the conditions you're doing. For example, let's say you're trying to make a drug. Do you know what its exact protonation state will be in biological solutions? Because if you're trying to do like docking, which is a computational modeling of like drugs binding to active sites, and you're using the wrong protonation states, the data is going to be totally wrong, right? Those protonation sites can do hydrogen bonding, which is a big effect, right? And then if there's an H there or not really matters, right? So the key problem here is that sometimes people draw things that in the wrong protonation state or in the wrong place, right? And then these wrong structures propagate into other documents, okay? And so especially I've noticed this, like if you look to go order a chemical, sometimes the way it's drawn is so bad that it's not correct. But here's one example. Diazo dyes are wonderful dyes that get used for a variety of things, including photochemistry, acid-base indicators, etc. This is methyl orange. You can buy it as the sodium salt. The sulfonate is deprotonated. It's this diazo group. And so it's sometimes called what's a push-pull, because this is an electron donating group, and this is an electron withdrawing group. Yeah, so people were studying these. And then I was helping a collaborator with some computational chemistry, and we were looking at papers, and I realized that sometimes in these papers, it was drawn like this, right, protonated. And sometimes the proton was drawn elsewhere on the molecule. And the pKa of this thing has been measured. It's about 3.8, okay? And so this is the issue, right? 3.8 is not this thing. Sulfonic acids are very acidic with pKa's in the negative region. So my favorite strong acid is tauxilic acid, because you can get the hydrate and weigh it out as a solid. And it's a strong acid, right? And that's a sulfonic acid. That's like this. So, so it shouldn't be a weak acid, right? So using chemical, like organic chemistry kind of intuition, where would you think this should be protonated, right? And I asked my OChem colleagues, and a few of them, you know, some people had different answers, right? Because everybody has different ideas and builds their intuition and knowledge differently. And so thankfully, the crystal structure is known. If you go and look it up, it's actually on the azo nitrogen. And it's only on the azo nitrogen next to the sulfur side. So here, the lowest energy I calculated by energy, and that matches the crystal structure. And then I checked the other sites, and they're all higher. In fact, the sulfonate that some people were picking is the most unstable place. You can also do some electron arrow pushing. So if you push from here to there, from here to there, from here to there, and then from here to the nitrogen as a lone pair, that makes sense as this being the most basic site. So just because someone throws a structure in a paper or a website doesn't mean it's correct. Sometimes they're shown in the wrong structure, the wrong protonation state, and those mistakes can propagate. So the lesson here is look up pKa's, know your pKa's, check structures, look for crystal structures, look for calculations, be sure that you're in the right protonation state when you're doing things that have acid-base properties. So that's kind of my spiel for today. That was about 45 minutes-ish. We all make mistakes. That's normal, right? We're humans, right? And mistakes get in the scientific literature. We should try our best to prevent them from getting into literature and try to fix them. And so it's easy to kind of get nasty, but we want to be nice to each other. We need to be supportive of science in general and scientists around the world. But we should be, critical of data and be sure that, okay, are we sure that the data is what it is? All right, double check things. The other thing is that like 1 or 2 papers out there on something is not enough for a consensus. Like those Jack's papers on pyridinium, right? Like you need many, many papers showing that it's reproducible for something to be consensus science thing. I hate it when there's an article that gets published and then news articles like science says this. Like, no, one article says that. That's not science. Keep trying multiple methods and tools to study things, right? And keep good records. It's not just one way to study things is not going to get you the answer. Check your own assumptions, be willing to change, right? It's okay to be wrong. That's part of learning, right? It's sometimes the first step in learning something new is being wrong about it. And so you got to let your kind of conclusions go sometimes. And again, reproducibility. So that's what I've talked about today. I'm working on some other stuff similar to the pyridinium. I saw another issue similar to pyridinium I'm working on publishing. So look out for a paper on that. It's a different molecule, but similar problem, misinterpretation of data. And then even like things in organic chemistry textbooks may potentially be wrong. And so I'm looking at some organic-y stuff too with that. So, more on that hopefully later. We'll see if those come to fruition in papers, but I think we all should be aware of that there are mistakes, the mistakes get into literature, and that we should try to, prevent that and fix them. So thanks for listening. Again, this is the scientist I am today is due to all the people I've worked with. So my academic advisors, my colleagues and peers, my collaborators, right? I couldn't be the scientist I am without them. My colleagues here at DePaul, know that I love to walk into their offices with papers I found. Like, you seen this? Is this legit? Like asking them questions about their field and stuff, because I want to learn more, right? And then all my DePaul research students, there's too many to list. And so they're the ones that I train and teach, and we do the work together. And then they get independent and go do the work themselves, and they're great students. And I learned so much from teaching them, training them, and working with them. And so Nick Ching did the computational work with me on the organic molecule, the organic drug that should, or natural product that should not exist. Mackie did the EChem on the pyridinium with me, and Josh did some pyridinium and acetonitrile stuff. And then all of this is pretty much funded by just startup money and department money. I haven't had any huge grants doing this, but we can get good science done at PUIs, primarily undergraduate universities. We're just a little slower. And again, thank you, Ben, for hosting this and running OSP. I think it's a great resource and a great idea and thing that you're doing. And then, of course, the science community, Chem Twitter, Discords that I've been on, and Blue Sky and so on, that folks that share their knowledge and help each other. So yeah, thanks for listening, and I'd love to answer or discuss any questions.
Appreciate it, Kyle. Thank you very much. Really, I enjoyed the pyridinium one, especially because I remember seeing some of those papers when it first, back around 2010 or so, and this pyridinium facilitates CO2 reduction, might be a way to do something with it, and then it kind of nothing ever really came of it, and I'd always kind of wondered what happened to that.
Yep.
So it's interesting that it does seem to work to some extent, but not at all the way that was proposed.
Yeah, there are people have been able to people have been able to observe methanol in very low yields, though. They haven't been able to get as high yields as ever were reported.
No.
Again, it seems very potential dependent and condition dependent. Like, what voltage that electrode needs to be at for how long to make it the right surface to do that is unknown. It's still at this point. There's some computational stuff out there that's pretty good, and some surface Raman and surface studies that people did, but it's so complex and hard to interpret that I'm not sure. I didn't show all the papers here. There's tons of them, but I kind of stopped following the heterogeneous surface chemistry stuff because I'm more interested in the homogeneous chemistry, right? So,
sure.
But yeah, that's the bottom line is platinum electrodes, man. Don't use them as inert electrodes to measure things. Use glassy carbon.
Definitely not. Yeah, that's platinum electrodes. Also, I was made aware at some point a few years back where people were using platinum to circuits, not as an inert electrode, but one they knew it could circuit really well. And then it turned out that in the course of the experiments, platinum was coming off of their cathode. It was coming off of it, depositing on the anode, and changing what was the actual chemistry at the anode.
Oh yeah, and, it's not inert. it does get oxidized. It does get, and so that's actually how cisplatin, the anti-cancer platinum drug, was discovered. A biologist was studying the effect of electrochemical voltage on cells, and he noticed that they stopped growing when he was doing it, but then it was only with a platinum electrode. And it turned out that with the platinum electrode, he was using an ammonium chloride buffer, and it made cisplatin in solution. And that was a thing that was causing, which is now a drug is used for cancer.
Yeah.
That was the thing that was causing the cells to stop growing, not the electrochemistry.
That's really interesting. I had never heard that.
It's cool.
It's incredible, the stuff that happens by accident sometimes.
Yes, that's how good science sometimes get discovered.
That's
so.
Really
are there
cool.
any questions from the peanut gallery?
Yeah, well, we got some in the meantime, I'm gonna take advantage and ask a couple more myself.
Yep.
So the electrochemistry in particular seems to be a ripe area for, again, not. I mean, I'm sure people fraud as well, but just it's easy to misinterpret things. Are there any other just general electrochemistry things that you come across with it that you kind of along the same lines? Don't make the assumption that it's a particular thing. Got a lot of information. Are there any other electrochemistry tips that you might have, just things to watch out for, common errors?
I mean, it's so sensitive to impurities, right? So be absolutely sure that you've excluded oxygen from your electrochemistry, right? Because oxygen can like I've seen people, especially when teaching students like early on, that, oh, there's a bump there. Hey, there's a reduction. No, that's just some oxygen. We didn't bubble. We didn't purge it with argon long enough, right? And the same thing again, like you've got to purify your solvents, purify your electrolyte. So I use tetrabutyl ammonium hexafluorophosphate, or TBA, I call it, for my electrolyte and organic solvents. And I buy the 98+% stuff, and then I recrystallize it from HPLC grade methanol and dry it in a vacuum oven right before I use it, because like any small impurity in electrochemistry can have a huge effect, because you're looking at this small surface when you're doing a cyclic voltammetry. So yeah, there's been plenty of other electrochemistry wackiness out there. And like I said, I'm looking at another one that actually was a 2022 paper that was also like the electrochemistry. They're interpreting something wrong. They must have had some acid in there because they used a platinum electrode, and sure enough, that same signal shows up. And they think it's something, and it's not.
It is.
So,
It's amazing how things keep.
yeah. So again, that's why you got to read literature, man. You got to get deep in there.
Very true.
Yeah.
Not seeing a lot of questions now, but I've got a couple more. On the computational side, so I've recently started doing computational fluid dynamics. Not the same as FT.
Yeah.
But I've learned very quickly that models, the model is trying to make everything work, and it is capable sometimes of sort of being something that is unrealistic, but will make force the model to converge or whatever.
Exactly. Yeah. You have to. So I've had that issue too with there's software you can buy, or there's some free one now where they'll try to model cyclic voltammetry and then tell you what's going on, but it'll force in parameters that don't make chemical sense, right? Or don't make physical sense, just to make the data fit. So once you have a good data fit, like check all your parameters and like, does that make sense?
Yeah,
right?
exactly. Yeah, the over parameterizations, especially heat spectroscopy.
Oh yeah.
You can have non-ideal capacitors, Warburg, you can make anything fit if you had enough parameters. Ultimately, that also reality.
Yeah, it's got to reflect reality, and it's got to be reproducible.
Exactly.
Yeah, and I feel like, and I've heard many people talk about this idea. There should be a like journal of reproducible experiments, where basically you just reproduce something from the literature and show it can be reproduced.
I mean, I feel like that might be popular.
Yeah.
just to confirm.
Yeah.
That's great.
Yeah, I mean, if somebody across the world does some science, then publishes it, I should be able to do it and get to the same conclusion, right?
Yeah, absolutely.
So
that's a big thing that's happened now. DOE and some national electrolyzers and things like that. Like they're starting to now. I think they've realized that you need to have protocols, not just establish targets, to actually make sure that being. They're starting to do like round robin testing. Got these 4 different labs. Here's the issues that we ran into. Here's how we solved it. Here's things. Error. 4 different groups, same measurements, but hardware it could still. I feel like I think people are starting to realize that big. It is a big issue, because papers that are brand new thing and done this great stuff. Then 5 years later, everybody, actually couldn't do it, and here's why.
Yeah, there was a blog for a while, again back in the 2010s, 2000s, called Blog Syn, SYN, and it was literally just like they were organic chemists working at various places, and they were anonymous for reasons of again that retaliation, but they would basically see a paper and say, hey, that looks cool, let's see if we can do it, and then they would like blog about their efforts trying to reproduce either successfully, and then they would report their yields and compare the yields versus what was reported. And sometimes things worked and sometimes things didn't. Right. And then they would try to troubleshoot why and contact the original author, and sometimes there was just something like some details that was just left off the experimental. It's crucial, right? Or something. So
it's a thing. So the last question I've got, and this is probably more out of ignorance than anything else, but in general, with especially like chemical synthesis and things like that, do you have any again, just kind of basic advice, stuff to watch out for, things like purify your precursors and things like that, but in terms of like let's say somebody's doing a synthesis, they think they have actually made something new, and like they've checked that
Uh-huh.
their precursors were good and all that. Um, how would you recommend somebody try and confirm that they've actually generated something, or maybe a new
Yeah.
way to do it, or whatever?
Yeah, exactly. So like especially for synthetic stuff, right? For usually you try to use multiple types of NMR and IR and the mass spec. But you have to kind of sit down and look at all the data and make sure it's consistent. So like in that case of those folks proposing a organic molecule that couldn't exist, like the mass spec, just because you run a mass spec doesn't mean you're going to see the primary mass. Sometimes, like depending on the conditions, it can react first, and so that's what happened. Is their mass spec was telling them something. They tried to fit everything onto that, but really in those mass spec conditions, in that mass spec, it had been dehydrated in the gas phase during the ionization, and so they were seeing the mass of it minus H2O mass, and so it was the wrong mass, right? So I see organic chemists use high res mass spectrometry to say like, oh, it is what we think it is, but they got to be careful.
Yeah.
And so check everything. And again, I kind of like, for me, it's like, okay, NMR, IR, mass spec, and then DFT and crystal structures. Like if you can get all of those things to agree, you're good. But if there's significant interpretive disagreements between those, you've got to think and check things harder. So yeah, it's again using multiple techniques. And I was talking with a friend who was talking about how he does it for his group when he's publishing papers. Is they literally print out all the spectra that for some new molecule, and they will pass it around to like several people and sitting at the table and say, without knowing what this is, interpret it and tell me what you think. And they make sure that everybody's interpretations are consistent. And if there's inconsistencies, then they discuss their interpretations and discuss the double check that the things are legit, right? And go re-examine it, right? So just to be like every spectra basically that they're putting in the supporting info, but it's really like what they're using to understand something has been kind of looked at by multiple people and interpreted by multiple people. Yep.
Like you said, especially for something like that, where potentially new thing that you've made and you want to actually have
Yep.
made that.
Yep. Yep.
Yeah, no, that's fantastic. Well, I appreciate it, Kyle. Thank you again
Yeah.
for being willing to give a presentation and giving a really interesting.
Yeah. Thanks.
Being able to reproduce work is a critical thing in all science.
Yeah.
These were great examples of how someone can unintentionally, again, like you said, there's a difference between fraud and just genuine interpretation.
Yep.
This is a great lesson. Try and
Yeah.
make things better.
Yeah. And I think also too, just knowing about this, right? Like
Mm-hmm.
when kids and growing up, and like we see media showing scientists being like, Eureka, I know this because of this one thing I did, and I'm the one,
Yeah,
and that's not how science is done, right?
that's true.
And also like they think like, oh, science is absolute and like objective,
Mm-hmm.
but that's not really true. And so being clear, it's one of the things I like training when I'm training my students, being clear about like what science actually is versus how it's kind of presented in non-scientific media. So
Absolutely, really good way to put it. Far too often, it's like somebody runs a single experiment on a TV show and they get an answer. Good.
yep,
Well,
yep.
it'd be great if
No,
that's
that's
how it
not how that
actually
works.
worked, but
Yeah.
no, that's fantastic. Thank you again, Kyle.
Thanks.
Great talk, and thanks to everybody for coming out and listening. Came in late on this. The recording will be uploaded to YouTube. Any final words, Kyle?
Um, well, if any folks want to chat more about this, or have thoughts or questions, you can always reach out to me. Email is kgrice1@DePaul.edu. I'm not hard to find. I love chatting and collaborating and just trying to make things better.
Excellent. Thank you again, everybody, and have a great weekend.
Yeah.